Hello, I'm Hope Rugo, a breast medical oncologist at the University of California, San Francisco, and Director of Breast Oncology Trials and Clinical Education. I am speaking at AACR 2024 in the Clinical Translational Trials section as a discussant for two very interesting trials that were done in the UK. These are called the partner trials, and what they explored was how to add a PARP inhibitor to chemotherapy in the safest and most effective manner in order to improve neoadjuvant response to treatment for patients with early stage breast cancer. But they also looked at patients who had germline BRCA mutations versus patients who had triple negative breast cancer and were treated in the neoadjuvant setting. So very interesting. So the first trial, which looked at olaparib in patients who had germline BRCA mutated early stage breast cancer, explored different scheduling. And the reason why they explored different scheduling is because it was known that giving olaparib with a taxane in the metastatic setting resulted in severe bone marrow suppression from a much earlier trial a decade ago. So they tried this gap scheduling where you might have primed with chemotherapy before giving the PARP inhibitor, in this case, olaparib, or you could give the olaparib first and then give chemotherapy. It turned out that the priming with chemo and then giving the olaparib a couple of days later seemed to make a really big difference in outcome and intolerance. And what they saw was what appeared in a very small hypothesis generating study, a remarkable improvement in the response. And then they looked at overall survival probability in terms of event. And the patients who had the gap scheduling, so starting chemo and then olaparib, had this remarkable response. So it's quite intriguing data. There will be more correlative studies that have been done, but this actually fits into preclinical data that was already done with the olaparib. So very, very interesting data. The trial with triple negative breast cancer is interesting because they use the same approach to understanding gap scheduling, either giving it before or after chemo, the olaparib, and they use the same olaparib after chemotherapy approach. But what they saw was no improvement in outcome in an overall phase 2-3 study of triple negative breast cancer. This trial, of course, was much greater powered. They had a lot of patients, but there was no difference in any of the efficacy measures adding olaparib to chemotherapy. They've collected a tremendous number of samples, so they'll be able to do a really exciting correlative work in a laboratory to try and see definitively whether or not there's a subset of triple negative breast cancers that might have homologous recombination defect or methylation defects that might render those cancers more sensitive to the use of olaparib. But overall, in the larger group, there was no benefit. So very interesting combination studies, tremendous work, investigator-initiated studies, and I'm very excited to discuss these at AACR.